All right, let's just jump right in. Welcome to today's Explainer. We are unpacking something absolutely wild in the AI news space today. A true game changer for artificial intelligence and medicinal chemistry. We're looking at the integration of OpenAI's GPT 5.4 with Molecule.one's Maria Lab system. Now, we've all heard a million times about AI writing text or coding, right? But Today we're looking at an AI acting as a near-autonomous chemist. We're talking about an AI literally, physically running and optimizing complex chemical experiments in a real-world lab. So it really begs the question, what actually happens when an AI can physically run its own experiments? Just imagine that for a second, an AI stepping out of the digital realm of pure reasoning and into the physical world. It's not just spitting out a formula on a screen. It's actively driving robotic arms to create and refine brand new medicines. It's sci-fi stuff. But to get why this is such a massive leap, we kind of need to understand the historical roadblock it's bypassing. There's a great quote from Newswise that hits the nail on the head here. They say, drug discovery is like molecular Tetris. Chemists snap atoms together, adjusting the pieces until everything fits. And honestly, just like Tetris, finding that exact perfect fit is incredibly stressful. Except here, it eats up massive amounts of time, energy, and capital. Chemists are out there constantly hunting for the exact right combo of catalysts, ligands, and substrates just to force molecules to behave in very specific ways. Now, you might be wondering why that's so hard. Well, a big part of it comes down to a concept called chirality, or handedness. Basically, a lot of molecules used in medicine exist as non-superimposable mirror images. Think of your left and right hands. They've got the exact same parts, but they don't overlap perfectly, right? In chemistry, your body reacts very differently to the left-handed version versus the right-handed version of a molecule. One might completely cure you, while the exact same molecule mirrored might be highly toxic. Without super precise guidance, traditional synthesis usually gives you a frustrating 50-50 split of these left and right-handed molecules. Chemists are obviously aiming for asymmetric reactions, trying to get, say, 95% of the safe, desired form, but getting there traditionally takes an agonizing amount of trial and error. Section 1. The Autonomous AI Chemist. So OpenAI and Molecule.one basically set out to build an autonomous AI chemist to solve this exact headache. Just think about the traditional chemist's workflow first. To optimize a reaction, they're manually designing reaction matrices, synthesizing reagents, running iterative tests, a process that easily takes months. But compare that to the autonomous AI pipeline. It's a continuous, fully automated system driven entirely by AI. It literally squashes months of grueling lab work down into mere days and does it at a fraction of the cost. Section 2. Inside the closed-loop pipeline. Okay, so how does the whole shebang actually work? It's a 4-step closed-loop workflow. Step 1. GPT 5.4 acts as the reasoning brain and generates a chemical hypothesis. Step 2. The Maria robotic lab takes that exact hypothesis and physically executes the reaction at a super tiny microliter scale. Step 3. Analytical instruments, things like mass spectrometry, measure the physical output. And step 4, those hard results feed directly back into GPT 5.4. The AI looks at that empirical feedback, learns from it, and boom, instantly generates the next hypothesis. It completely replaces human guesswork with rapid robotic iteration, fixing reaction drift totally on its own. Section 3. Solving the Chan-Lam puzzle. To really put this to the test, researchers gave the AI an open-ended prompt. And you know what it did? It independently chose to tackle a notoriously tricky chemical reaction called the Chan-Lam coupling. For context, this is a standard method for forming carbon-nitrogen bonds, which are absolutely everywhere in pharmaceuticals. But it's famously stubborn, often giving really low yields, especially with a substrate called primary sulfonamides. The AI spotted this as a high-value target all on its own. It drafted up proposal OAI M103, suggesting they toss in mild oxidants, specifically one called TMPO, as an additive. The AI's theory was that TMPO would stabilize the oxidative conditions and finally fix those historically low yields. And while the results were absolutely stunning, by adding tempo and letting the closed loop do its thing, the AI boasted the mean reaction yield across tested substrates from a baseline of 16.6% to an impressive 25.2%. Now, if you're in process chemistry, you know that seeing a mean yield jump like that across over 80% of tested substrates is just a massive, massive win. And the data really highlights the real-world impact of that optimization. If we look at the frequency of highly successful reactions, meaning they pass the 30% yield mark, the baseline conditions only hit that 15.6% of the time. But under the AI's optimized conditions, that number soared to 37.5%. It literally more than doubled the frequency of high-yield reactions during that microliter scale screening. And the best part? It wasn't a fluke. When human teams ran independent validations at a larger scale, 11 out of 14 tested substrate pairs showed higher yields, with most seeing a straight-up twofold increase.
Section 4. Predicting with sparse data.
Now, if you follow AI news, you know these models usually need mountains of data to learn anything. So how did GPT 5.4 pull this off? It actually relies on a sparse data approach. Gathering massive datasets from experimental chemistry is just wildly expensive and slow. This system skips all that by using low-cost physical organic parameters. Instead of needing millions of data points, it specifically models the transition states during a chemical reaction, the radical capture, the intermediate phase, and the reductive elimination. By understanding the actual physics of how the catalyst and substrate interact in those split seconds, the AI can accurately predict outcomes and even transfer that knowledge to totally unseen reactions. It's incredibly efficient. Section 5. The future of AI labs. So, what's the big takeaway from all this? If you actually want an AI running your lab, there are 3 absolutely critical integration points you need to get right. First up, hardware and analytical feedback. GPT 5.4 is brilliant at reasoning, sure, but it is totally blind without ultra-reliable robotic liquid handling and pristine automated data pipelines. Even a little noisy data will wreck the iterative loop. Second, prompt engineering. You've got to set strict chemical boundaries, safety constraints, and target metrics. Prompt too broad, the AI wastes time. Too narrow, you kill discovery. And finally, robust independent validation pipelines. You have to remember, AI proposals are just experiments. You still need cross-functional teams of human chemists, machine learning engineers, and roboticists to run validation batches before scaling up. Automating the loop doesn't erase the human element. It just puts the path to validation into hyperdrive. Which brings us to a pretty profound question to wrap up this explainer. We've clearly proven that automating the loop accelerates the path to validation. But are our physical labs actually ready to keep up with the speed of AI? As our digital reasoning models evolve at lightning speed, upgrading our real-world scientific infrastructure to match that pace is definitely going to be the next great hurdle. It is an absolutely fascinating space to watch. Thanks so much for joining me today. Keep asking questions, keep learning, and I'll catch you in the next explainer.